All right, we are going to start in on muscle physiology. This tissue is designed to contract. That's its function. This happens to be a image drawn by Leonardo da Vinci, who studied human anatomy by doing dissections of human bodies and drawing them. So in a multicellular organism, we have physiological systems, and many of them depend, in fact, on muscle tissue, the specialized ability of muscle to contract. Muscle can do that because it contains non-soluble fibrous strands or filaments of polymers of actin and myosin, which can interact, and these are called thin for actin and thick filaments that are made of myosin. So the filaments are made of polymers, multiple units of monomers of actin or myosin, strung out into a non-soluble filamentous structures, either thin filaments or thick filaments, that can interact by sliding past each other. The three major types of muscle tissue include skeletal muscle, cardiac muscle, and smooth muscle, which differ in a number of their characteristics. Terminology is a little bit different for muscle tissue in that the plasma membrane is called a sarcolemma, and the cytoplasm inside is called the sarcoplasm. All of them, this tissue is excitable tissue. It can be triggered to contract by the spread of action potentials along the membrane surface. Some of the characteristic features of the three types of tissue are shown here. In terms of skeletal muscle is, of course, attached to bones and involved in movement of the skeleton. There aren't any skeletal muscle cells after birth. During the embryonic development, skeletal muscle cells fuse into multinucleated skeletal muscle fibers. So we don't talk about cells, we talk about and we talk about skeletal muscle fibers. These are quite long, in some cases many centimeter long multinucleated fibers formed by the fusion of individual skeletal muscle cells during development. They are long, multinucleated, unbranched fibers. The nuclei are pushed, multiple nuclei are, because of the fusion, they're multinucleated, so the nuclei are pushed to the outer edge of the cylindrical shaped skeletal muscle fiber. They are voluntary. The activation of this tissue is voluntary. It is controlled by the somatic motor division. So efferent output from the central nervous system to activate this muscle is consciously or voluntarily controlled. Involves the somatic motor division. Signal comes from motor neurons that send out action potentials, and they then send a signal over to the skeletal muscle fibers. Cardiac muscle is different. It has single nuclei. The function, of course, is to pump blood. It's found in the heart. The key typical histological characteristics are that it is branched, and you can see the single nucleus here. There's a single nucleus and this branching-like structure of that cardiac muscle cell. If you look carefully, it's fairly obvious in the drawing, and if If you look carefully at the photomicrograph, you can see that it also is striated or striped, just like skeletal muscle. At the ends of the branches are structures called intercalated discs, and they allow for connection, at least a connection in terms of allowing for ion flow from one cell to the other, from one cardiac muscle cell to the adjacent cardiac muscle cell. So these structures, intercalated discs, are found at the ends of the branches. You can see them fairly clearly in the photomicrograph. They're all over the place, and you can see them here in the drawing at the very ends of these branches where one cardiac muscle cell meets another. The activity is non-voluntary. This tissue, similar to smooth muscle tissue, is controlled by the autonomic nervous system, and I remind you that the autonomic nervous system has two divisions, the sympathetic division and the parasympathetic division. So unlike skeletal muscle that's controlled by the somatic motor division, this is controlled by the autonomic nervous system. Somatic nervous system versus autonomic nervous system. Yeah. Okay, in terms of skeletal muscle tissue is uninucleated. It has a single nucleus, uninucleated cells that are unbranched. They're long, kind of reasonably long, nowhere near like skeletal muscle fibers, but reasonably elongated cells. They are activated by a non-voluntary system. The autonomic nervous system signals them. There's no obvious striations on them. Let's move on. Okay, here we see a actually extremely nice photomicrograph of a series of skeletal muscle fibers that are lined up parallel to each other. What you have are the multinucleated skeletal muscle fibers. All of these nuclei that I'm encircling here are all associated with this single skeletal muscle fiber and are at the surface. Inside you see that very repeated clear pattern of striations all through here, very clear repetitive pattern of striations. This kind of tissue, when compared to cardiac muscle, tires relatively easily. It's responsible for body movement. The muscles are attached to the bones by tendons. It is also quite adaptable. It will increase in size, not number. The skeletal muscle fibers will increase in individual size. Not the number of them, but the size of them will increase with increasing usage, which is what causes muscle tissue to get bigger with exercise. Similarly, if it's not used, you can get muscle atrophy where the fibers get smaller, and that can, some of you may have experienced that when you had maybe a cast on your arm and you didn't use the arm muscles and it became atrophied, visibly smaller once the cast is removed. Okay, so it is a striated tissue, as I told you. These reflect the very, very regular arrangement of actin and myosin filaments. The dark line in the striation is actually where the filaments are overlapping with each other, and therefore that's more dense, so it's darker. It is not made of cells, as I said, but fibers, multinucleated fibers that are formed by fusion. The entire muscle, when you look at it, is made up of multiple bundles of fibers held together by connective tissue. Essentially, if you look at a single muscle, and you can see we're talking about the whole muscle. And by the way, each muscle is considered a unique organ on its own, because it is quite separate. It itself is a collection of different tissue. So each muscle is a separate organ attached to the bones by tendons. You have, if you look at it, inside the muscle, you have these bundles of muscle fibers, quite a few muscle fibers bundled together. And then if you look at a single bundle, which happens to be called a fascicle, okay, a bundle of muscle fibers is called a fascicle, which I believe is spelled like this, fascicle. So that's a single fascicle there. There's another fascicle there, you see. So this single fascicle is made up of multiple muscle fibers. So there's an individual muscle fiber, extremely long, multinucleated, cylindrical structure. Once you get into that and you look at the single muscle fiber itself, it is made up of multiple myofibrils. And here you see a single myofibril. And you see the molecular arrangement of the thin and thick filaments. The blue in there are in the single myofibril are the thin filaments, and the red ones are the thick filaments. And the dark stripe causing the striations of this tissue are where the two overlap. So in this area here, there's overlap, so it would be a darker stripe. In this area, it would be a darker stripe. And in between, where they don't overlap and you only have thin filaments, that would be the lighter striping, the area of lighter striping. So the diameter is fairly small, 10 to 100 micrometers. The average red cell is the average diameter of a single red cell, a red blood cell, just to give you something to compare to. The diameter of a red blood cell is about 8 microns. That's a micron or a micrometer. Okay. So you have these myofibrils that are bundles of thin and thick filaments. Initiation of contraction occurs by triggering through a stimulus, action potential sent out from a motor neuron. So these efferent signals are sent out by motor neurons down the axon, and they meet the at the end of the axon you get splitting of the term into multiple different terminals or areas that innervate, join to, in other words, that innervate multiple muscle fibers. The meeting point where the actual motor neuron terminal of the axon meets the skeletal muscle fiber is called the neuromuscular junction. Neuromuscular junction, and I'll show you in the next few slides what that means. So again, what we have is here a single motor neuron, and it's sending out an axon that innervates a number of different skeletal muscle fibers within a particular muscle. So here you see this motor neuron is innervating these two muscle fibers. This motor neuron is innervating these three muscle fibers. And they're not, the muscle fibers that are innervated by a single motor neuron are not going to be all close, really close to each other. They're going to be sprinkled throughout the muscle. Just want to point out to you that this is a cross section through the spinal cord, and the central area, this butterfly shaped central area, is shown here, is the gray matter. It's where the cell bodies are of the different types of neurons. All the motor neurons are found down in these areas here. So the cell bodies of the motor neurons are found in these areas, and they send their axons out to innervate skeletal muscle fibers. So this idea of motor neuron triggering off the sending out a signal that activates the skeletal muscle fibers involves action potentials. If the motor neuron is stimulated enough, an action potential is signal is okay, is sent down the axon of the motor neuron and can innervate as many as 100 separate muscle fibers within a muscle. And that arriving signal to the ends of the axons, to the axon terminals, will trigger the release of the neurotransmitter acetylcholine. There is a space between the axon terminals and the skeletal muscle fiber. They don't actually touch each other. There is a space, and it is called the synapse, okay, or synaptic cleft. It's a synapse. On the other side, where the synapse, where the axon terminal comes in, on the other side, the area on the sarcolemma, in other words, the cell membrane of the skeletal muscle fiber, is called the motor end plate. So the acetylcholine that gets released by arrival of a depolarizing action potential, it'll trigger the release of acetylcholine. This is the abbreviation for acetylcholine. It'll trigger acetylcholine release, which diffuses across to the motor end plate and binds to specific receptors, acetylcholine receptors on the motor end plate. So here you see a axon, and these are the axon terminals, and this action potential arrives down. This is a Schwann cell that surrounds and insulates the axon, but the axon, the signal arrives down the axon onto a skeletal muscle fiber, and here we have a larger diagram, or a higher magnification diagram, showing that axon terminal here at in this area here at the lower left, this diagram. So what's happening is the signal arrives, and that triggers entry of calcium into the axon terminal. It causes the opening of calcium channels, which allow calcium to move in, and that in turn, and all of this is written on the next few slides, and that entry of calcium in turn will trigger vacuoles that contain, or vesicles rather, that contain the neurotransmitter acetylcholine to move to the membrane at the axon terminal at the at the neuromuscular junction, and release their contents of acetylcholine. The acetylcholine will drift across onto the area called the called the end plate. All of this area of the skeletal muscle fiber here within the synaptic cleft is called the motor end plate. On that motor end plate, what you have are these receptors, which are also acting as channels. So when the acetylcholine binds to the receptor, the channel opens and you get sodium and potassium can pass through. Remember, cells, or you should know that cells, all, in fact, not just excitable cells like neurons or muscle, but all cells have calcium-potassium pump action going on all the time. So normally, sodium, did I say calcium, magnesium? it's sodium-potassium pumps working all the time. So normally sodium is pumped out of the cell by the action of the sodium-potassium pump, and potassium is pumped in. And these ions, sodium, potassium, are pumped against the concentration gradient. This is a pump action that requires some energy. So sodium concentration outside of the cell is building up. It's higher than inside because of the sodium-potassium pump, and potassium is higher inside than outside because of the sodium-potassium pump. If a channel opens, because sodium is higher outside than inside, sodium rushes in. And some potassium also can go out. So you get these sodium-potassium channels opening and sodium enters, and the depolarizing effect, the effect of making the cell less negative of the positive sodium ions, it causes depolarization that spreads over the surface of that motor end plate. If the signal is strong enough, it'll spread even further over the entire surface membrane of the skeletal muscle fiber. That skeletal muscle fiber membrane is called the sarcolemma, and the cytoplasm is called the sarcoplasm. It is the membrane, the sarcolemma, is continuous with a system of tubules that extend down into the sarcoplasm. It's sort of a sac-like structure, somewhat akin to the endoplasmic reticulum. There's these tubular structures, T-tubules, that join up with the sarcoplasmic reticulum. I'm sorry, it's the sarcoplasmic reticulum is somewhat analogous to the endoplasmic reticulum in cells, except here it's a storage site for calcium. So if you have a signal arriving from an axon that innervates onto a skeletal muscle fiber and releases acetylcholine, it opens up channels on the motor end plate that allows sodium to enter. You get a depolarizing effect, and that depolarization on the membrane spreads down these structures here, these T-tubules. Let me show it in a different color. Here, these T-tubules spreads down. These T-tubules are shown in orange or yellow from the surface. There's the surface. So a signal will spread from a motor end plate and then go down, depolarizing signals, and will have an effect on the sarcoplasmic reticulum, this blue structure that is a storage site for calcium. The effect is the release of calcium into the sarcoplasm so that calcium can interact with the contractile filaments of the muscle. This is not a bad slide, actually, showing this kind of effect. Let me just change this. So you have that neuromuscular junction, the meeting place of the axon terminal and the skeletal muscle fiber. There's the T-tubule here shown in green in this diagram. Sarcoplasmic reticulum shown in yellow this time, not blue like the last slide. There's a myofibril, the plasma membrane, which is called the sarcolemma. I'll talk about sarcomere later. Okay, so an action potential arrives, causing depolarization of the axon terminal, release of the neurotransmitter that drifts across and binds to the receptors in the motor end plates that act like channels. The sodium goes in. A lot more sodium rushes in, and you get a depolarization that spreads outwards away from the motor end plate, and then outward away from the motor end plate, and then down. Let's go for another color that makes it really clear. Away from the motor end plate, and then down the T-tubules, and triggers release of calcium from the sarcoplasmic reticulum. This is another diagram showing the same idea here. Again, the axon terminal releasing acetylcholine binding to receptors, triggering a depolarization, and that spreads across and then down in just a second. Okay, the effect, as I said earlier, is that the action potential moves down the T-tubule and affects the sarcoplasmic reticulum to cause calcium release, and calcium then has an effect on the myofilaments to allow them to interact and slide past each other. In fact, what's actually going on is that the calcium binds to troponin, part of the actin filaments. So the thin filaments have troponin on them, and that changes the shape of the troponin. Actually, no, it's not quite that way. What happens is that a structure called tropomyosin is covering the troponin. And when you have calcium arriving, the tropomyosin gets pulled away and reveals on the actin filaments myosin binding sites. So now myosin on the thick filament can bind to actin on the thin filament, and it binds and moves it along, just like two strands grabbing, pulling along, releasing, grabbing, pulling along, releasing. I'll show that to you perhaps more clearly soon in a later diagram. So you get this repeated cycling of actin-myosin binding, release, and sliding along. And that is what causes the muscle fibers in general, the whole muscle fiber structure, to contract. And that will continue until signals stop arriving from the motor neurons and the calcium is no longer being released. And that's when the muscles relax, when the signals stop arriving, the excitation signals that come from the motor neuron down the axon. When the muscle stops contracting, calcium then gets pumped back into the sarcoplasmic reticulum, and that allows for repolarization of the membrane of the skeletal muscle fiber. The skeletal muscle fiber goes back to being more negative. It is repolarized and ready to contract again if a new signal. This kind of mechanism of binding and releasing and ratcheting along is shown here. It is not exactly intuitive, but it's not the binding of, this is a myosin head here. It's not the binding of the myosin head to the troponin on the actin filament that is the energy requiring step. It is the release of the myosin head. So when that gets released and then gets bent forward like that, that's what takes some energy. That's what requires ATP. Remember, all contraction requires energy to be available, and that energy is in the form of ATP. So what you have is you have calcium released from the sarcoplasmic reticulum, binding, as you can see here, it's binding to the troponin, but that causes the tropomyosin to move away. The tropomyosin is this long strand shown here. It's a winding strand winding around the molecule, and it reveals, in that way, it reveals the myosin binding sites. Here, these myosin binding sites, all these round yellow circles that are present on the actin are the myosin binding sites. With revealing of the myosin binding site, you get binding of the myosin to the actin. and then that moves along like a ratchet system. So it slides, it attaches and then bends in that manner, and these slide along in this direction and that direction, opposite directions to each other, so the overall myofibril is contracting. Then the next step is that it has to release, and that requires ATP. It releases, and that's shown here. It releases, and then the head of the myosin head moves back and is ready to bind again. This is another diagram describing the same kind of thing from a different textbook, actually, because I thought that maybe between the two it would be clearer. It's not exactly intuitive how that system works, but it involves binding. They've changed the order here. When ATP binds, the head releases. ATP is hydrolyzed, releasing energy. The head pivots, binds, and then moves along. Okay, it's the release. ATP binds, head releases. It's the release of the myosin head from the thin filament that is the energy requiring step. Now, in terms of the smallest functional unit within the muscle, it is the motor unit. I think I'm going to stop here, and then we'll continue later with part 2.